The second part to this video is gustation. Gustation is chemical coding that evolutionary scientists believe was the first sensory system of the earliest animal before the neocortex forebrain evolved. There was areas of the midbrain and hindbrain devoted solely to sensory. The stimulus for gustation is organic substances contained in chemicals that are perceived as sweet, sucrose, sour, HCl, bitter, quinine, salty, NaCl, and umami or meaty, MSG. A combination of activity in 5 kinds of receptors along with smell give perception of the taste of food and meat. And as you can see on the tongue, at the top here is the papillae, and here are some taste buds. Our tongue is arranged so that we taste things in the order of bitter at the top, sour second, salty third, and the tip of our tongue tastes sweet things. And our taste buds are up here. This is more of a blown up image of the papillae. These can contain about 100 or more cells of receptors for food or taste, as opposed to the eye or auditory systems who only use maybe 3 to 5. Okay. Taste buds are contained here in the papillae, which is here also. Taste receptors, which are here and here. Have excitable membranes and release transmitters to excite other cells. Taste receptors are generated and replaced every 10 to 14 days, which is why food may taste better after an extended fast or being sick or having an illness. Taste receptors are located inside the taste buds, which are here, and it shows is a bigger picture here. The papillae of the tongue are where the actual taste buds are here, and the blown up picture here. And this is the inside of the taste receptors. There. This is transduction of gustation. When a substance tastes salty, for instance, it means that the saltiness receptor has detected the presence of sodium. Ions cross the membrane and produce an action potential. Sweetness, bitterness, and umami receptors resemble the action of metabolic receptors and therefore activate G proteins, molecules, and second messengers within the cell. Then transmission starts. Although each kind of taste bud is detected by one receptor, several receptors create a particular fire pattern that is perceived as a single taste experience. So that means that all of these things make up one taste that is sent to the brain. Information from the posterior tongue and the throat travel along bunches of cranial nerves 9 and 10. I don't have a picture of those nerves, but they are located around a person's face and through the back of their skull. Taste. Nerves project to the nucleus of the solitary tract in the medulla. From the NTS, the information branches out to the pons, the lateral thalamus, and the amygdala and the ventral posterior thalamus, finally terminating into the areas of the cerebral cortex. The sensory matter sensory cortex is where the touch or food texture is detected on the tongue, and the insula or primary taste cortex is where taste is perceived. Innervation is ipsilateral in the sensory modality. The sensation of taste can be affected by the culture and family values, but also be genes and hormones. Give an example from the textbook. The genetic differences affect taste, also how hormones affect taste. And taste buds, age, hormones, women's sensitivity rise and fall during pregnancy, and hormone cycles.